Ashika from 2nd BSc Microbiology, and I am here to explain gene transfer mechanism. The transfer of genetic material from one cell to another cell by a bacteriophage is called transduction. Transduction has been found to occur in variety of prokaryotes, including certain species of bacteria. Example: Escherichia, Pseudomonas, Salmonella, Staphylococcus, etc. The bacteriophage containing the bacterial DNA is called transduced DNA. Transduction is a process of genetic transfer between bacteria mediated by viruses. Was discovered by Norton Zinder and Joshua Lederberg. They demonstrated this phenomenon using Salmonella bacteria. Generalized transduction. In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called the bacteriophage or phage. The phage attaches to the bacterial cell and injects its nucleic acid into host cell. A phage enzyme is produced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and phage code proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material. Specialized transduction. Specialized transduction involves a transfer of only a few specific genes from one bacterial cell to another by means of a phage, the lambda phage, which infects E. coli is a well-studied example of a special transducing phage. When lambda phage infects E. coli, the phage DNA enters the cell and then integrates into a specific site on the host chromosome. When an E. coli culture carrying the lambda phage is induced, phage particles are produced. On rare occasions, a piece of bacterial DNA, for example, the gall gene near the specific site of insertion, remains attached to the phage DNA and a piece of phage DNA is left behind the phage. They develop are defective because they do not carry the entire phage genome but can still infect other cells. The defective phage can attach to another bacterial cell and the DNA can be injected, but both phage and bacterial DNA now become integrated into the new host chromosome. Only bacterial genes located near the site of integration of the phage DNA can be transduced, hence the term specialized transduction.